Friday harder. I mean, there's so many options. It was just, it was hard for me in both ways at the same time. It's exposed to so much material.
So hello, my name is Daniel, and today we're going to talk about comparative analysis of phenotypic sequences. And I just want to start by describing what a phenotypic sequence is. And this is an example that I like a lot, which is courtship displays in birds of paradise. So the idea is that we have behavior varying over time, and this is discretized. So each one of these boxes are modules, and within them we have different units of behavior. So the idea, we have a phenotype with different states that varies in a gradient of time. So another example that we can think about is morphology. So with the dental arc of these fishes here, we have different types of teeth. Teeth is a serially homologous trait. And we have different shapes and different functions associated with different positions of this morphological sequence. So the sequence that I'm going to So one thing that's cool is that talking about animal behavior, morphology, or molecular sequences, that's what we're used to. There's lots of attributes that all these traits or molecular sequence share with each other. And today I'm going to talk about a sequence type of trait, which is the calling songs of male grillos crickets. So here what we have is some sonograms, my main focus today is going to be in the temporal pattern variation. So what I'm focusing is in regions that are silent and regions that we have the sound. The sound, of course, are these black lines. And one important thing is that all these crickets produce the sound the same way. So the morphology of the wings that they rub to produce the sound and the behavior is homologous. So this is important when we're doing this type of work. It's a fundamental hypothesis of homology. So just an overview of questions and how we're going to try to do this is that the things that we are interested in is how rates of evolution vary along the sequence trait. Another important question is, are these rates correlated among the sequence? And finally, do we see evidence for context-dependent variation in the rates of evolution on these traits? So in the case of our work, the context is, do we see difference in rates associated with silent regions of the song and chirp or sound regions of the song? So the first step on trying to do this is to get a phylogeny. Here is already different from molecular data. We are going to estimate the phylogeny using molecular data. It's not the trait that we're using, right? Second, we need an alignment. So working with a sequence, we need to have a hypothesis of homology among the species, and that's what the alignment is going to do for us. Here, again, it's not molecular sequence, so we are going to condition the alignment on the phylogeny for the species, right? And then after we have the alignment, we can go and fit the models that we are interested in to answer our questions, right? Okay, so the first thing that we are going to do here is to transform this sonogram into a type of data that our models are more adequate to work with. So to do that, we are discretizing the time variation into states. And the states are silent states in gray, and then we have chirp or sound in black, and then we have unknown. We have unknown because the time length, the time period that we discretize, if we see both sound and silence, we just say that we do not know what that is. And that's why we have unknown states bounding each chirp or sound. So the first problem, the first challenge here is the alignment itself. First is that repetitions are a big source of alignment uncertainty. So here, for example, we have two sequences with repeated silence states. We can create multiple alternative alignments for these sequences, and they are it's really difficult to tease those apart. So ideally, what we want is to compute the probability of those alignments, given that we observed the tree for those species, right? One framework that we can do that is Bodify, but another challenge is that Bodify is expecting us to give molecular sequences. So we need to put some tricks such that the hypothesis of similarity of the states that we do is reflected on the model that we are using. But after that, we do get a posterior distribution of alignments that we can use to integrate this unknown variation of the alignments. Another framework that we can use is to do distance-based alignments. And here's a little bit more easy because we can not use, instead of using the default distance matrices for molecular data, we can build our own distances, which is a reflection of our hypothesis of difference between the states that we observe on the sequence. And MAFT, luckily, have this implemented already. This is just a screenshot of the website, and you can use this option to deal with non-biological sequences. So this is how the data look like. And the first thing to look is that we do have lots of repetition all over the place. We have two types of chirps under our mapping of the sound, which is continuous chirps, or these chirps that are separated by much shorter regions of silence. And here is, of course, the phylogeny for the group. Another important detail here is that this is a recording over 5 seconds. So the first thing that we did is to try to get the alignment for the complete sequences. And this is an example of how it looks like. So yeah, it doesn't look that great. And so first thing is that, why I'm saying that is because what look like our working hypothesis of homology of these chirp sound regions change along the alignment. So at some point it's like this, other points are, we see other species with sound that is isolated. So we step back a little bit and try to recognize a more integrated view of homology of this. And by doing this, we thought, well, we can consider here repeated motifs of this calling song. So this is what we define song bout. So now song bout is a chirp of the constant one or of the one divided by small silences, separated, bounded, flanked by relatively longer regions of silence. So when we use this type of the song bounds to make the alignment, then we do get our gaps again because the sequence length is very different. So that's included another challenge on the whole work. But now we have a more consistent hypothesis of homology for the chirp regions. Although this guy here, Grillo's integer, creates a lot. Well, it's difficult. Let's just stay with it's difficult. So which type of model that we can use with this type of data, right? So to start with, we have discretized the traits. So we have 3 types of traits, and one of those are the gap states that were produced by the alignment. And here we have an interpretation for the gap states, which is the loss of the trait. So loss of the trait is different from silence. Because silence, we do have a observation that the crickets were not doing the behavior at that particular moment. A gap is the transition, the evolutionary transition that end up with sequences, bouts of different length. So it's a loss. So to deal with that, we can use a simple Markov model, and the simpler one would be just to have equal rates among all these states. And we define another one that we call the deletion model, which is transitions to the loss of the states along the sequence have a different rate. But one of the questions is how these rates vary along the sequence, and to deal with that, we are just using the same approach that we use for molecular sequences, which is to use independent gamma distributed rates vary along the sequence, right? So this is the same as the plus G, like GTR plus G, this is the gamma. But we need to incorporate autocorrelation between the sequences, so how we deal with that? When I say autocorrelation, what I mean is that if you get 2 sequence positions like this, 1 and 2, we expect that if they are correlated, then transition rates for the first position are very likely to be similar or equal to the neighboring positions, right? So to do that, we use a bivariate gamma distribution. So what it means is that we are sampling at the same time the rate for the first position and the second position that's neighboring it. And this is another really good paper by Young in '95. This model is not as used as the independent gamma model, but is also defined from molecular sequences. And here what we have in the left, we have an independent gamma distribution. These are just samples from the distribution to show how it's supposed to look like, the expected rate variation. And the one with correlated rate scaling values, so a correlated bivariate. The idea here is that under the correlated distribution, we are more likely to have similar rates for these two positions. Okay, so I'm going to show some results under this model. So first one is with the whole sequence, and what we see, and these are, so this is the alignment, and this line here is the model averaged rates of evolution for each of these positions. First thing is that we do find support for autocorrelation along these sound sequences, and you can see base rates with a bunch of spikes in different places. Now we have a similar type of model when we consider bound sequences. So we do have base rates and then a spike on the rates, but now more concentrated to the intership silent regions. So when we ask the question, do we see context-dependent evolution of rates on the sequences? Then to do that, we get the model average rates. We separate between the chirp-heavy and silence-heavy regions, and we do find a difference with silence seeming to be evolving and transitioning faster, evolving faster. So just to conclude here, first thing that is very important is that sequence organization is a way that evolution have to partition the traits, create integration and modularity, such that these different parts of a dental arc, for example, can evolve, different teeth can evolve to have different functions. And this is a very interesting idea, in my opinion. The second part is that I do think that this a sequence organization is an underexplored axis of variation in multivariate data that could open a lot of great avenues for interesting questions. And just to conclude, it's interesting that silence seems to be evolving faster than sound, but if we think a little bit, when the cricket makes sounds, it attracts predators and parasites. So maybe evolving silence is an axis of variation that's less constrained than evolving sound. And I just want to thank Theo, Jeremy, and Tony that help us with all the cricket questions. So that's it. Do I have 2 minutes, I think?
Yeah, I think we can go. Yes, you have 1 minute.
What?
Until the turnover.
So you should come. Questions? Yes?
Was there a lot of difference in sort of likelihood of different alignments? It would seem that many alignments would be almost equally likely.
Yeah, we do. So we do have regions on the likelihood, and my expectation or my hope is that we can sample well enough from those regions and incorporate them when we are estimating the model. That's it.